Okay, awesome. I see we have 100 plus participants, so I'm sure others will keep rolling in, but I just wanted to start out and say hello and welcome to everyone. Thanks for joining us for this New York Invasive Species Awareness Week webinar series. Before we dive in, we just have a couple of quick introductions and logistics to cover. So this event is part of the New York Invasive Species Awareness Week. This is the 9th year of celebration. And we wanted to thank you for being here to stay informed and involved in invasive species issues. Be sure to visit nys.info/NYISA for more info on events in your region this week, including a screening of the documentary Uninvited that's happening at 7 p.m. tonight. But today, for this afternoon, we're really excited to have assembled this panel of experts to give you the latest on the science and management of harmful algal blooms and their intersection with invasive species issues. This event is co-hosted by the New York Invasive Species Research Institute and the New York State Water Resources Institute. My name is Justin Dalaba. I'm the communications coordinator with NISRI. We're based at Cornell University, and our director, Carrie Brown-Lima, is here. She's going to give you a quick introduction of our role, and then she'll turn it over to Brian Gramlich-Ram, the director of the Water Resources Institute. And after they've given their introductions, we're going to pass the baton over to our experts. I just want to add one other thing. If you have questions throughout, please type those in the Q&A box, and we'll do our best to respond to as many of those as possible. We'll have some time for discussion at the very end. So with that, I'll turn it over to Carrie Brown-Lima.
Thanks so much, Justin. Are you seeing my screen and hearing me okay?
Yes, looks good.
Great. Well, welcome everybody and happy Invasive Species Awareness Week. It's such a pleasure to be here today with this great lineup of speakers. And this event is really in line with the NISRI mission, which is to work at the interface of invasive species research and management, and to communicate and coordinate invasive species research to help prevent and manage the impacts of invasive species in New York State and beyond. We were established back in 2008. And we're based here in the Department of Natural Resources and the Environment at Cornell University. And we're funded through the Environmental Protection Fund that's administered by the New York State Department of Environmental Conservation, actually. And the PI of the Institute is Dr. Bernd Blassi. I just wanted to take a minute in case some of you aren't familiar with all the great work that's being done here in New York State through the New York State Invasive Species Network. Here in New York, we are lucky to have a really progressive and comprehensive group of organizations and agencies that are supporting invasive species efforts. Here in New York, we have an Invasive Species Council that's comprised of 9 agencies that work with invasive species here in New York and have projects or programs related to that. We have an advisory committee. It's called the New York ISAC. It's made up of 25 NGOs or academic institutions, trade groups, and others that provide advice to the council. We have 8 regional partnerships for invasive species management. So if you're located here in New York State, you are placed within a PRISM, as we call it, that's the acronym. So do seek out more information on your PRISM if you want to get involved more locally in invasive species efforts. We have an IMAP invasives database, which is has a role to keep track of all the invasive species and depends on people like you to collect data on where those species are and enter other information about treatments and other management activities that are going on. And the role of the Research Institute is to work with all this network and to connect relevant research and improve the scientific basis of invasive species prevention and management. And we also look to understand research needs on topics such as harmful algal blooms, so we can bring together researchers to do that, share that information, or conduct relevant research. So just to, in a nutshell, we work in 5 main areas. We're trying to understand basically who's doing what and where. We facilitate information sharing through webinars such as this one, through conferences. We hold an invasive species track at the Cornell Cooperative Extension annual in-service. We also identify research needs and priorities from our stakeholders in New York State, and then we coordinate research to meet those needs and promote collaboration among different academics and manager groups. And we're also always seeking innovation and cross-discipline solutions and really trying to advance our knowledge on invasive species in our decision-making. I just wanted to point out we have a lot of resources available on our website and in our newsletter. We spotlight a researcher every month. We summarize relevant research that comes out in the journals, and we make it into a bite-sized format so that you can get a heads up on the research that's happening without diving into the journal themselves. We also compile all relevant research articles from about 30 journals every month, so you can check out the abstracts and know what's being published out there. We also promote and announce new information, new webinars, new conferences, funding opportunities. So with that, I just wanted to encourage you to, if you're not already on a PRISM listserv, to join our NISRI listserv so you can get monthly information such as this. Check out our website for additional resources, including recordings of webinars such as this one. And also, of course, feel free to reach out to any of us individually, including myself, and here's my email. So thanks so much for the opportunity, and looking forward to hearing more from our speakers today. So with that, I'll pass it over to Brian.
Awesome. Thanks, Carrie. Hi, everybody. My name is Brian Rum. I'm a director of the New York State Water Resources Institute, which is also here at Cornell. We have a general mission to identify and address the critical water resource issues for the state of New York. And we do that through research, outreach, and training, both here at Cornell, but also more broadly with other academic institutions, NGOs, artists, community groups, municipalities. So we have quite a network of folks who are in the right places at the right times to help us address water resource management issues. Every year it's quite a challenge to figure out what should we be working on. But what I'll say is, year in and year out, harmful algal blooms and aquatic invasive species are two of the things that we hear from. We hear about from a lot of different stakeholders, whether that's elected officials or community groups or researchers. And so it's great to see the intersection of these two issues getting addressed at this panel. And I'm looking forward to what the speakers have to say. Thanks, Justin, for putting in the link to our website. I would say if anybody has questions about what WRI is or what we do in this space, please check that out. And again, thanks to NISRI and some of our other partners, in particular, folks at the New York State Department of Environmental Conservation and at USGS for helping round out some of the important stakeholders that do this work in New York. So thank you.
Awesome. Thank you, Brian, and thanks, Carrie, for those introductions. So without further ado, I'm going to introduce our first speakers. And while I'm doing that, Jennifer and Rebecca, if you want to go ahead and start queuing up your presentation. So we have first up for you, we're going to give an overview of harmful algal blooms and invasive species. And you'll be hearing from Doctors Jennifer Graham and Rebecca Gorney. Jennifer Graham currently serves as the harmful algal bloom coordinator for the USGS Water Mission Area. She also represents the USGS on the Interagency Working Group on Harmful Algal Blooms and Hypoxia. She's a nationally recognized expert in cyanobacteria and associated toxins. And for over 2 decades, Jennifer has conducted research on the environmental factors influencing the occurrence of cyanotoxins in the United States. She has conducted both regional and single system studies at a variety of spatiotemporal scales. And following her presentation, you will hear from Rebecca Gorney, who is a research scientist with the New York State Department of Environmental Conservation's Division of Water at the central office in Albany. She manages the state's harmful algal bloom monitoring program and a statewide lake volunteer monitoring program. Rebecca received her PhD in natural resource management from the University of Vermont and has been with the department since 2015. She's worked in New York City's urban forests, Hawaii's rainforests, and alpine Colorado lakes, quite a range of ecosystems. And her recent work at the DEC includes expanding the agency's efforts on HABs research, mitigation, and outreach. So Thank you both for being here, and the floor is yours.
Great, thank you, Justin. Can you hear me and see slides?
Yes, looks good.
Excellent. So good afternoon, everyone. And, thanks to Justin and the institutes for putting together this webinar. We're really excited to be here this afternoon. Rebecca and I are going to start off the webinar by giving a little bit of background on harmful algal blooms and a high-level overview of connections with invasive species. And then the other speakers in the session will delve into those connections in a little bit more detail. We wanted to start off with just a basic definition of an algal bloom. And this isn't as easy as it may sound. There are a lot of complexities that go into algal blooms and harmful algal blooms, that starting with definitions is often a place where we start. Part of the reason for the complexity is that there are many different types of algae that can cause algal blooms. And we wanted to be clear right off the bat that algal bloom causing taxa are generally not considered to be invasive species. In marine environments, often it's the diatoms and dinoflagellates that cause algal blooms and harmful algal blooms, and in freshwater environments, it's most often the cyanobacteria. And cyanobacteria will be the main focus of the presentation today, although you will hear about some other taxa that cause harmful algal blooms in freshwater environments. So when we talk about a definition of an algal bloom, this definition is really subjective, and it often depends on who is defining and why they are defining. So if we're talking about aquatic food web studies, that definition might be very different than someone who is working on public health protection. We also want to kind of emphasize right here at the beginning that not all algal blooms are harmful. Some algal blooms are actually very beneficial and serve as the base of aquatic food webs. But the common definitions, the ones that you most often hear and the ones we most often use when we're talking about harmful algal blooms are an event where you have very high cell densities of algae in the environment. And actually what we mean by high also depends on the particular organism that is causing the bloom. So even within that high cell density, there's often a range that we'll use. Another definition that's used is dominance by a single or just a few species. Typically in a balanced ecosystem, you'll have very diverse communities, and when you start to see just one taxa dominate, that's indicative of a bloom of that particular organism. And then finally, another one that's used is just a visual accumulation of algae. If something if there's enough algae accumulated that you can see it by the naked eye, it's an indication that you may have a bloom. And the example that is on this slide here is a cyanobacterial bloom in the western basin of Lake Erie that is large enough that it can be viewed by satellite from space. So that's algal blooms. What makes some algal blooms harmful? Again, there are a lot of different harms that can be associated with an algal bloom. A lot of times we focus on the human health concerns. Both the freshwater and the marine taxa of algae can produce toxins that are potent enough to cause human illnesses and in rare cases, deaths. There are multiple routes of exposure to these toxins, including recreational activities, drinking water, foods, aerosols, and skin contact. And again, those vectors of exposure often depend on whether we're talking freshwater or marine environments. A lot of our research and a lot of the harms when we talk about harmful algal blooms are really focused on this human health perspective. However, there are other harms that go along with algal blooms when you have such a massive accumulation of biomass. And those include ecological health, things like degradation of water quality, alteration of aquatic food webs, and mass mortalities of fish, birds, and mammals. And then there are economic and aesthetic concerns that go along with these accumulations of algae or algal blooms that result in fish kills like the one that you see here depicted on the slide. And that can include decreased tourism and property values, commercial fishery losses, and increased drinking water treatment costs. So there are a lot of different harms that may result from an algal bloom. Again, it's important to remember that not all algal blooms are harmful, and not all harmful blooms are necessarily toxic. So there's a lot of complexity baked into this issue of harmful algal blooms. One of the things that we're most often asked as research is what causes an algal bloom, or what causes a harmful cyanobacterial bloom. And there are a lot of interacting factors that influence the occurrence of algal blooms, a lot of interacting factors that influence the potential toxicity of a bloom. And some of them are the same and some of them are different depending on what organism we're focused on and what particular toxin we're focused on. But this array of interacting factors includes nutrients, it includes water clarity, it includes hydrology, climate, weather patterns, and rainfall and runoff can have important influence. And biological community interactions also have a really important influence on the potential occurrence of harmful algal blooms. And those interactions include invasive species. And Rebecca will touch on this in a little bit more detail here in a few minutes. Harmful algal blooms are considered to be a relatively new. First report of a toxic cyanobacterial bloom was in Australia in 1878, when a bloom was linked to livestock deaths. Shortly thereafter, animal deaths associated with cyanobacterial blooms were reported in the United States. There was an incident in Minnesota in 1882. And these events have, they've occurred fairly routinely since then. And this timeline here really just shows reported first reports of animal mortalities associated with cyanobacterial blooms and cyanobacterial toxins. And you can see that these were reported every so often. And we have, this documentation through 1990. There has been an apparent increase in events in the late 90s into the early 2000s, and it became a lot more difficult to keep track of these reports, first reports since then. There was a major mortality event in Iowa in the 1940s. There were reports that it was thousands of migratory birds, wildlife, livestock, and domestic pets died after exposure to a bloom in a particular lake in Iowa in the 40s. And this incident really was the impetus for some of the foundational research associated with cyanobacterial bloom and toxin occurrence in the United States. It was the impetus for the first known recreational monitoring program in the United States, as well as some early mitigation studies. And so this is an issue that has been with us for a long time. These early studies really, some of the results of them have really stood the test of time and have resulted in a solid foundation on which all of our science has been built since then, including that cyanobacterial blooms are an issue that can sometimes, but not always, be toxic. The events usually, but don't always, occur during summer months. High nutrients and other ideal ecological conditions result in blooms. And there's even a list of potential taxa of concern that was developed coming out of these studies in the 40s and early 50s. At that time, the one thing that they couldn't do was actually identify the toxins that were associated with the blooms. They knew that they were complex organic substances, and they were almost impossible to determine. And so since that time, we now have the identify to quantify and identify what these compounds are. And there's been a lot of great work since that time. I'm going to turn the presentation over now to Rebecca, who is going to talk about harmful algal blooms in New York State and some of the wildcards that affect harmful algal bloom occurrence, including invasive species. Rebecca.
Thank you, Jennifer. So each state kind of has developed its own approach to documenting harmful algal blooms. In New York State, we use a platform called HABs, the New York Harmful Algal Bloom System, which is built in Esri ArcGIS Online tools and is a place for folks in New York to look at where blooms have been reported recently. And we retain a list of all the blooms that have been reported throughout the year. And users can filter by lake name or county. And most of the reporting is based on visual evidence of a bloom occurrence rather than extensive testing, because you can see by this image, we get a lot of reports and we are not capable of sampling every single one. The visual indication is considered sufficient. Next slide, please. So we've been documenting HABs for 10 years and the story is complicated. The first graph on the left shows that each year we find a few new water bodies that never had a HAB documented before. So we are, have a steady growth in the total number of locations where they've been observed. You can see on the map that they are statewide in nearly every county and every type of water body and all different land uses, all different, soil compositions, they're everywhere. The bottom slide shows, however, that our reporting efforts have really leveled off. We have each year about the same number of lakes are reported with HABs year on year on year. So the last, well, they were increasing in the beginning, but the last 6 years have been very stable. Next slide, please. There are, the basic recipe that Jennifer mentioned, high temperature, high nutrients, that we expect harmful algal blooms to occur. But when you introduce invasive species, those can change the fundamental ecology of a lake. One particular invader of note is zebra mussels. However, this could be applied to mystery snails or new zooplankton or new fish. They can really change how lakes function, particularly Dreissenid, quagga, and zebra mussels can affect nutrient recycling, how nutrients are moved through the lake, especially at the bottom areas near the sediment interface. And Dreissenid mussels can also change the substrate of the bottom of a lake. Then when you add in the factors of climate change, everything climate change does, leading to higher temperatures, big storms, etc., tends to favor cyanobacteria. And then we also have, of course, new contaminants coming into lakes that are receiving all of our runoff. And we don't know exactly what new emerging contaminants, such as pesticides and personal care products, we don't exactly know which of those can affect HABs, but it is likely that they do. Next slide, please. One example of an interaction between invasive species and harmful algal blooms is a new type of cyanobacteria that was recently found in the southeastern United States growing on hydrilla. So hydrilla is an invasive plant that tends to spread aggressively, and there are several different subvariants of hydrilla. This particular cyanobacteria produces a neurotoxin that then tends to bioaccumulate and affect birds, mammals, reptiles, etc. This differentiates it from other cyanotoxins, which we have studied more recently that only have mammalian endpoints and do not have bird endpoints. So this new food web of cyanobacteria, hydrilla, and a neurotoxin is concerning. We have not yet documented it in New York State. Our invasive species unit, led by Kathy McGlynn, occasionally does send samples of hydrilla in the isolated locations where hydrilla is found in New York, and we have not detected the cyanobacteria yet, but it is a good example of how these two topics intersect. Next slide, please. So while Jennifer mentioned that cyanobacteria have been around for a long time, there's obviously some changing parameters with invasives and climate change and whether harmful algal blooms are getting worse or more toxic or how they're moving around in the environment is still somewhat unknown. The exact recipe of drivers that will lead for a bloom to occur are not predictable. They don't follow modeling tools to have a predictive model very well. The health effects of the cyanotoxins that we know about and the emerging cyanotoxins and exactly how toxic they are, potentials to humans or pets or other animals is also an area still with lots of gaps. The movement of cyanotoxins through the water column and potentially impacting drinking water use or food impacts through irrigation, et cetera, is also under study. We have looked in New York at several emerging mitigation strategies, such as the photo on the bottom right, which is an ultrasonic, solar-powered ultrasonic buoy that we have used in a couple of lakes in the state. And then here today, you're going to hear more from our next speakers about the influence of invasive species. So next slide. I think that's it for the two of us, and it was a pleasure. We worked together quite a bit, and it was a pleasure to do this talk as a team. Thank you.
All right. Thanks so much, Rebecca and Jennifer, for that awesome overview of what's going on and helping set the stage for the conversation here. It's really interesting to see how the number of algal blooms across New York State have somewhat leveled off in recent years, and also hear about some of these emerging areas of study. So next, we're going to hear about a case study or case studies of invasive species and their intersection with harmful algal blooms. Unfortunately, our next speaker could not be here today, but she was generous enough to pre-record her presentation for us. And you can defer any questions to Jennifer Graham. She will try to help answer some of those for us. But next, you're going to hear a pre-recorded talk from Dr. Mary Ann Evans. So she is a research ecologist at the USGS Great Lakes Science Center based in Ann Arbor, Michigan, where she contributes to the restoration and conservation science branch. Her current research focuses on drivers and consequences of harmful and nuisance algal blooms, including nutrient, light, and Dreissenid influences on benthic algal blooms, cyanobacterial harmful algal blooms in Western Lake Erie, nutrient and algal dynamics in other river-influenced nearshore areas of the Great Lakes, and translating harmful and nuisance algal bloom science for diverse audiences. Prior to joining the USGS, she studied hypoxic dead zones in the Chesapeake Bay and Gulf of Mexico, and she received her PhD in biology from the University of Michigan, studying phytoplankton ecology in Arctic lakes. So this is Mary Ann Evans' talk.
Today, I will be presenting about case studies of invasive Dreissenid mussel effects on Great Lakes HABs. Specifically, I will be presenting on cyano-HABs in Western Lake Erie and on nuisance benthic algae, such as Scledophora, in the Great Lakes nearshore. Western Lake Erie cyano-HABs represent the largest HAB area in the Great Lakes. This bloom occurs each summer, varying in location across the lake, generally starting midsummer and reaching into September or even October, depending upon the weather. It has led to drinking water disruptions, the largest of which, the Toledo drinking water crisis in 2014, interrupted drinking water supplies to Toledo as well as many surrounding areas, almost 500,000 people, for 2 days during the height of the summer. There have also been smaller drinking water disruptions to individual wells or to small drinking water supplies around the basin. Because of this and other human and environmental impacts, the Western Lake Erie HAB has become a binational research and management focus. All of this leading to a current attempt to reduce nutrient loading from the basin into the western basin of Lake Erie, with a focus on spring nutrient loads in accordance with the Annex 4 nutrient management plan. I'm going to touch on two aspects of the interactions between this harmful algal bloom and the invasive Dreissenid mussels. This is not to discount the importance of nutrient management, the importance of weather, and also controlling this HAB. It's generally thought that the Dreissenid mussels have increased the propensity of the system to produce harmful algal blooms. And I'm going to, as I said, touch on two aspects of that. The first is termed the nearshore shunt, an idea that the mussels, by filtering out algae and releasing nutrients into the water, increase the cycling rate and decrease the cycling distance, the distance that nutrients move through the water before they are cycled through the system again, in such a way that it holds more nutrients near shore, where they can lead to algal growth. And in the Western Basin, essentially the entire basin operates as a nearshore area due to its relatively shallow depth. And there's some citations on here for where you can find the research and theory that goes into this. The second aspect is a direct feeding interaction between Dreissenids and algae. Dreissenids are filter feeders and use their intake siphon to pull in water, including the algae that it contains. They then filter out those algae and reject some of them as pseudofeces or by pushing them back out through their intake siphon if it, for example, starts to clog. It's been found that Dreissenids selectively feed on some algal species over others, and Microcystis, the bloom-forming species, as well as other colonial cyanobacteria, are frequently among the rejected species. This consumption of other species frees Microcystis from competition for nutrients, for light with those species. Again, the references are on the bottom of the slide, and I've sort of depicted this in the cartoons on the side where we have algae of two species. For example, a green oval algae would be a more desirable species, and the colonial globs of Microcystis in the lighter green. And then my cartoon art description of a Dreissenid mussel. As the mussel feeds on the algae, it's selectively removing the desirable green algal species and has not removed proportionally less of the Microcystis colonies. That then, even if all of the algae are growing at the same rate, in this case doubling between these two panels, results in a community that is very dominated by Microcystis. And it is thought that this, operating in the large lake, makes the lake more susceptible to turning the available nutrients into the harmful or undesirable algae. The second vignette that I'd like to present for you is looking at Cladophora, which is a shoreline fouling benthic algae. It usually grows as part of a community. So you can see in the image on the left here, there's strands of Cladophora, but also some other algae of different colors, different lengths mixed in there. And this Cladophora biomass growing on the bottom can lead to it releasing late in the summer due to wave action or high temperatures, and then washing up on shore where it can create, as in the image on the right, large mats of decaying algae. They're unsightly, they stink. They can harbor harmful bacteria, which can lead to beach closures if it's a bacteria that could cause human illness, or can lead to bird deaths if it's a bacteria that can lead to bird botulism. So this is considered a nuisance algae, can lead to environmental harms, and again is a focus of international attention because of its interaction with nutrient loading from the watershed. So to dig into a bit more of what controls Cladophora biomass, and thus Cladophora washup. Like many algae, Cladophora is in the Great Lakes generally limited by phosphorus availability or other nutrients. We'll let phosphorus be a stand-in here. It's controlled by light availability, And like Cladophora and other attached benthic algae, it needs a hard substrate to grow on the bottom. All of these, however, have interactions with invasive Dreissenid mussels. Those mussels, as we talked about for the planktonic HABs, can increase phosphorus and nutrient availability by increasing the cycling of those in the near shore. They increase light availability by filtering the particles out of the water column so that the light can penetrate deeper into the water. And just by nature of having hard shells, they can increase substrate availability for the algae. And all of those individual linkages have been the focus of various studies. I have some examples here. There are many more out there. These studies have mostly been at sort of individual site levels or a stretch of coastline. And so my group is currently conducting research on a large-scale assessment of Cladophora so that we can ask if the site level patterns hold up at the large scale. We're doing this using a combination of sentinel site work with divers, as you can see in the top image, where they're using quadrats to, in a detailed way, assess both the Cladophora, the other algae, and the Dreissenid mussels on the bottom, as well as assess the available substrate. And we combine this with nutrient samples, with measurements of the physics of the water column, so that we can assess both Cladophora availability, Cladophora overgrowth in the various systems, as well as the drivers of that growth. We're pairing this with an AUV assessment so that we can cover more area, essentially. The divers, as you can tell from the image, are looking at one very specific location in a lot of detail, but we can't cover the whole bottom of the lake that way. So we are training a computer vision algorithm using some of the divers' images and samples to be able to use photographs of the bottom collected by an autonomous underwater vehicle, which is the lower photo on the side. And that AUV can then cover more of the bottom and get us information about not everything that the divers assess, but the coverage of Cladophora across a wider area, and put our sentinel site assessments in context. The research community is then using models to combine these and many other types of data and information to predict the Cladophora outcomes across a broader range of the Great Lakes. So this produces a lot of data. We only have a short time today, so I just want to present some of our ongoing work comparing sentinel site and AUV work. The data here are from our Waukegan transect in southern Lake Michigan. It's circled on the map, and our other sites are also shown for this sentinel site project. The individual maps show an area about the size of 3 football fields around our 6-meter and 10-meter sentinel sites. And the color intensity shows the Cladophora percent cover at those sites. The sentinel site is indicated in the little hexagon in the center, and 4 replicate samples taken at that site are shown above the graph, so you can get an idea of the nearby variability. The larger area is the percent cover as determined by the AUV, and this shows a high local variability in that coverage, thus putting in context the more detailed work we're doing at the individual quadrat level. Our next step in this work will be to look at estimates of substrate availability, hard versus substrate, and Dreissenid mussels across this larger 3 football field sized area, and compare the Cladophora percent coverage with the available substrate and see how those distributions match up. In summary, today I've highlighted several ways that Dreissenid mussels impact cyanobacterial HABs and benthic cladophora across the Great Lakes through impacting light and nutrient availability with their filter feeding behavior, by directly feeding on and selectively feeding on algae in a way that can change the community composition of the phytoplankton, and by providing substrate to benthic algae. Here are the citations from all of the studies that I referenced today. I'm sorry I was not able to join the webinar in person. Jennifer Graham has kindly offered to address questions during the comment period. And if you have additional questions, you can feel free to reach out to me through the webinar organizers. Thank you.
Wow, so interesting to be able to visualize some of those interactions with invasive species and learn more about the tools and technologies we're using to better understand these complex dynamics. So next, now that we've covered the overview and set the stage, learned about some of the science, now we're going to hear more on the management side. And so next up, we have Dr. Steven Souza, who's going to present. He's the owner of Clean Waters Consulting LLC. He's also the founding partner of Princeton Hydro LLC, serving as president of the company from its inception in 1998 until his retirement in 2019. Over the past 35 years, he's dedicated his career to the management and restoration of aquatic ecosystems, in particular lakes and reservoirs. Dr. Souza is a past president of the North American Lake Management Society. He's an adjunct professor at Temple University and an instructor and course coordinator for the Rutgers, New Jersey Agricultural Experiment Station Office of Continuing Professional Education. So thank you for being here, Stephen. The floor is yours.
Well, thank you very much, Justin, and thanks to our previous speakers for basically setting the stage for me. So we're going to talk a little bit about the consequences of HAB management and whether or not in doing so, we're swapping out one problem for another. Now, we can look at lakes in a very simplistic way, and there's theoretically, we can essentially characterize lakes as either being macrophyte or weed dominated, or conversely, algae dominated. And this all goes back to studies that were conducted by Dr. Steve Carpenter in the 1970s and early 1980s. But essentially, if we have a weed dominated or a macrophyte dominated lake, it tends to have clearer water, because most of the productivity is sediment based. We have plants that are growing from the sediment. There's less water column productivity. Conversely, algae dominated lakes, again, this is a very simplistic way of analyzing this, but they tend to be more turbid, less clarity, less water clarity. And that's because most of the productivity that's occurring in those water bodies occurs within the water column itself. So this illustration basically shows two water bodies side by side, one being algal dominated and the other being weed dominated. But in reality, we know that lakes are really a little bit of both. We have both weeds and algae occurring in common. And the thing is, though, we know also that eutrophication, particularly accelerated eutrophication, tends to push lakes very quickly into an algae-dominated state. And when we talk about harmful algae blooms, we're really talking about an algal-dominated state that's really an extreme example of algal dominance. But in this case, it's cyanobacteria. And as Dr. Graham pointed out previously, cyanobacteria are bacteria, but they do photosynthesize. So they're photosynthesizing prokaryotes, but at the same time, they're very algae-like in many ways. Bottom line is that we do know we have a lot of tools in the tool chest that can be implemented to help temper or even prevent harmful algae blooms from occurring. And those restoration actions are designed to really try to bring the lake back into a state of balance where we do have some algae, we do have some weeds, but they're coexisting in a more balanced, healthy way. And the problem with HABs, again, both as all of our previous speakers noted, is that they do create some very severe impacts, much more severe quite often than the good algae. We have the toxins that we have to be concerned with, which are harmful not only to humans, but various other life forms. We see, as was discussed previously, that they can alter the food web of aquatic ecosystems. And more on a societal basis, there's lost recreational opportunities and higher costs to manage those water bodies, whether they're being used for recreation or for potable water. Just in the past 10 years alone, there's been over $10 billion spent in algal management and HAB management alone. And this is just to reel in some of those problems that are associated with harmful algae blooms. So again, this is a little bit simplistic, but as Dr. Evans pointed out previously, there are a lot of drivers, but we do know that one of the primary drivers of harmful algae blooms is the availability of a lot of bioavailable phosphorus in the water column. So one of the typical solutions that are utilized to reel in and try to manage HAB impacted lakes is what I refer to as putting a lake on a phosphorus diet. That is decreasing the amount of bioavailable phosphorus, whether that phosphorus is originating from the watershed or it's from some internal source. And the bottom line with that is that as we decrease the amount of bioavailable phosphorus, the amount of food, that decreases the potential for algal-based primary productivity, which also decreases the opportunity in most cases for harmful algae bloom, a cyanobacteria-dominated bloom. And because macrophytes, although they do obtain some of their nutrients from the water column, they're obtaining the majority of the nutrients, the majority of macrophytes from the sediment. So this sets the stage to then shift the lake back potentially into a macrophyte-dominated type of system. And in addition, competing for nutrients, we also have plants and algae competing for light. And so as we decrease the amount of turbidity in the water because of algal productivity, going back to that original slide, we increase the amount of clarity. That increases the opportunity for macrophytes to grow, particularly early in the season. And overall, we shift the lake back into a weed-dominated steady state. Overall, this tends to be a good thing. Most weed-dominated lakes tend to have better aesthetics and better recreational usage. They tend to have less of the eutrophication-related water quality impacts. They tend to provide better habitat and therefore fish, and therefore have a more robust fishery. There's less sediment disturbance because the sediments are maintained in a more stable fashion because of the root and rhizome systems of those plants. And beyond that, we find that weed problems actually tend to be easier to control than algae problems. Again, there's more tools in the tool chest to deal with weed problems as opposed to algae problems. Also, there's some allelopathic effects that are associated with certain plants. And what that means is that there's some plants that actually produce chemical compounds that have been shown to inhibit cyanobacteria bloom formation. I've provided you with a couple of citations, but these are based largely on lab experiments that have confirmed that certain species do elicit this allelopathic impact that does decrease cyanobacteria bloom formation. And unfortunately, some of those species are very invasive, and some of them are non-native species, such as Eurasian water milfoil in hydrilla. But as Dr. Graham pointed out, it's not a perfect world. There are examples now of AVM where we have this deadly waterfowl disease that's associated with a toxin linked to a certain cyanobacteria that actually grow on hydrilla. So it's not a perfect world, but there are certain plants to keep in mind that as they begin to dominate a given habitat or given lake or reservoir, that they will generate some of these allelopathic chemicals that in themselves can help decrease the potential or the severity of a cyanobacteria bloom. So are we swapping one problem for another? Well, the bottom line is that algae control often results in more weed growth. If we have clearer water, particularly in the spring, that tends to be advantageous for the growth of a number of different plant species, but especially some of the early season invasive species such as Eurasian water milfoil, curly leaf pondweed, hydrilla, and parrot feather. So could HAB control lead to an invasive species dominated macrophyte community? Yes, that's possible. And in some cases it's likely. However, it's not all that bad because as I mentioned before, even though we're potentially swapping one problem for the other, there are a lot more ways to control those weed problems. Far easier to do, far easier to predict when we're having the onset of a macrophyte problem, and far easier to manage than cyanobacteria problems. And whether we're talking about harvesting, grass carps, systemic herbicides, contact herbicides, benthic barriers, or dyes, these are all tools in the tool chest that Dr. Lubnow will talk about in more detail. So in summary, lakes, we can look at this in a somewhat a simplistic manner and understand that they tend to oscillate between a weed-dominated and an algae-dominated steady state. But for the most part, weed-dominated lakes tend to have clearer water, less water quality problems than you see with algae-dominated and particularly cyanobacteria-dominated or impacted lakes. So although we may be swapping one problem for another, weed control is easier to implement and weed problems are easier to predict, control, and manage. So with that, I'm going to turn it over to Dr. Lubnow, who will close out with a discussion about some of these techniques.
All right, thank you, Steve. Hey, can everyone see that?
Looks good, Fred.
Thank you. All right, so yeah, I'm going to continue where Steve left off, talking about invasive plant management and the interrelationship with HABs, harmful algal blooms. So like Steve mentioned, are we swapping one problem for another? Well, as was discussed, lower nutrient loading into a lake means lower amounts of algae in the water column. So it tends to be clearer. Sunlight hitting the sediments, you will have more plant growth. And yes, some of that plant growth might be invasive species. However, these plants are not producing cyanotoxins. Plants also produce some habitat and refuge and stabilize the sediments. And like Steve mentioned, the submerged aquatic vegetation, the SAV, can be, not always, but usually is easier to manage than a cyanobacterial HAB. And again, I want to emphasize with the HABs, there are a lot of health issues, beach closures, concerns over pet deaths, all associated with cyanoHABs. And this is just showing you an example of a lake-wide bloom at Lake Kapakon, as opposed to if we had high densities of submerged aquatic vegetation, it tends to be limited in the shallower areas where you have sunlight hitting the sediments. Like Steve mentioned, these large-scale HABs, very frequently, they're very difficult to get a handle on. They're very difficult to predict unless you have a lot of water quality and watershed data. So I'm going to go through the list that was previously provided and just talk a little bit about the interaction between SAV management and HABs. The first is mechanical weed harvesting. So essentially, you're removing the plant biomass. You're removing phosphorus, which stimulates HABs. And we've done some studies to show that some aggressive mechanical harvesting can remove, up to 12 to 15% of the phosphorus targeted to be reduced on an annual basis. And removing that SAV can potentially provide habitat for benthic algae, although, from our experience, we have not seen a large shift from plants to benthic algae as a result of mechanical harvesting. It's very difficult to see that in the field. So, and a large part of that is mechanical harvesting. You are selecting specific areas. It might be to open up some habitat for fish. It may be to open up areas for recreational use, but usually you're not harvesting an entire water body. Again, you're removing the biomass, so that's very effective in removing nutrients as well, but we generally don't recommend mechanical harvesting if there's an early infestation of an invasive species. If you have something that was just identified, such as water chestnut here, there are some alternative techniques to remove that. For example, manually or physically removing the plants by hand in an early phase of an infestation is a lot more effective, because when you're doing harvesting like this, many of the invasive species like Eurasian water milfoil, hydrilla, they grow primarily through fragmentation. So if you're harvesting in an early phase of an infestation, you can really end up resulting in distributing that invasive species. So, and this is just showing you an alternative to mechanical harvesting. This is a truxer. And this unit is a lot smaller, and it's suited for shallower areas between 0 and 3 feet in water depth, very different than those large paddle wheel mechanical harvesters. So again, just to go back to manually removing the plants, if you have an initial infestation identification, it can be far more effective to physically hand remove that plant. You're removing phosphorus and nutrients and biomass on a larger scale, and there's minimal chance of triggering a HAB event associated with manually removing plants. Sterile grass carp have been shown to be very effective at controlling high amounts of submerged aquatic vegetation. It's a permitted activity, requires some permits. The fish are somewhat selective in terms of the plants that they will feed. But what I do want to emphasize here is overstocking can really trigger a HAB event. I'll give you an example that about 4 or 5 years ago, we were hired by a lake community in Virginia. They used to have hydrilla, and then they stocked sterile grass carp. They completely eradicated not just the hydrilla, but all the native vegetation, and they ended up having problems with HABs. When we spoke with them, we found out that they ended up stocking 4 times more the amount of sterile grass carp than what we would have recommended, because in states like New York, Pennsylvania, and New Jersey, you stock based on vegetated acre. They stocked based on total acreage. So they stocked far more carp than they needed. The carp ate all the hydrilla, but ate all the aquatic vegetation as well. And that triggered a HAB, because as the carp are feeding, they are excreting nutrients, and they are also stirring up the sediments and making habitat available for HABs. So again, carp can be effective at controlling invasive plants or aquatic plants in general, but you really have to be careful on the stocking. Contact herbicides, which are one of the most common ways of dealing with aquatic vegetation. The bottom line is contact herbicides are relatively low in cost. It's a permitted activity that requires a licensed applicator, but they do have varying levels of toxicity on non-target organisms. And you have to keep in mind with a contact herbicide, you have to have enough biomass, and that product is killing the plant from the outside. And as it's breaking down that plant, it's making nutrients available, and not just nutrients, but organic forms of phosphorus. And the cyanobacteria are actually better adapted at using organic forms of phosphorus than other algal groups. So doing large aggressive treatments with contact herbicides do have the potential to produce a HAB event by mobilizing and making some of this organic forms of phosphorus readily available. The alternative to a contact herbicide is a systemic herbicide, for example, like Sonar. These products tend to have a low toxicity to non-target organisms. And what's nice is you can put in a concentration low enough to take care of an invasive like Eurasian water milfoil or hydrilla, but at the same time allow the native vegetation to grow. They also have a longer lasting effect than the contact herbicides, because the systemic herbicides, the plants are taking it in through the roots, and at least in the case of Sonar, they're shutting down the plant's ability to produce chlorophyll, so they essentially starve. But these products are expensive compared to contact herbicides. You need to be careful with flushing because the product needs to be in for an extended period of time, but there is a lower risk of triggering a HAB. You're not mobilizing nutrients very quickly over the summer season with systemic herbicides. So even though the chances of having a HAB are lower with systemic herbicides. The product is a lot more expensive than contact herbicides. There are also benthic barriers. It's a more of a proactive strategy of preventing plant growth through the season. You're laying down a specific type of material that's gas permeable, so it allows gases to be exchanged from the water and the sediment. Can be relatively easy to install. It is a permitted activity, and unless if it's not maintained, you could easily have a layer of benthic algae growing on the surface. So typically what you do is you lay the barrier down, and then at the end of the growing season or the following spring, you do have to clean it off and then reinstall it or re-anchor it. It tends to be most effective for property owners that are concerned about their dock areas, and also a lot of golf course organizations will use benthic barriers to prevent the development of submerged aquatic vegetation. Dyes, which the dyes are primarily designed to limit algal growth. They do not kill the algae, but in some cases, these dyes are still a permitted activity. They have varying levels of effectiveness in controlling submerged aquatic vegetation. They're really more designed for algal control. They will give the water a bright blue color. It reduces the amount of available light for algal growth. But usually where we see organizations or groups using the dyes tend to be very small ponds or at golf courses. And then finally, I do want to mention drawdowns. Drawdowns can be done for a number of reasons, for example, dock and shoreline repairs or restoration. But drawdowns have the potential to control submerged aquatic vegetation, but it's highly dependent on the species. So species such as hydrilla or Eurasian water milfoil, both invasive species, can be controlled through drawdowns. Others, such as the pondweeds, including curly-leaf pondweed, can actually increase after a drawdown. And winter drawdowns are the ones that are typically allowed. Again, a permitted activity, but their effectiveness is strongly dependent on the weather. I mean, if you want a real good, effective level of control, you need at least 30 days of sub-zero weather, and you really need freezing conditions before you get the snow. That's really important because the snow can actually insulate the plants. So the winter drawdowns really, they have the potential to control the SAV. Summer drawdowns are not recommended. They could easily trigger a HAB event. When you lower the water level, you're increasing the temperature over the sediments. You are also lowering the potential of that water body flushing. And so when you have that water body with a lower flushing rate, that can stimulate cyanobacterial growth. So you can actually exacerbate or trigger a HAB if you're doing any sort of summer drawdown. So generally, most state organizations don't allow summer drawdowns unless it's an emergency action. Winter drawdowns are the ones typically done to control at high levels of SAV growth. So, in conclusion, there is no magic bullet for any in-lake management option, whether you're trying to control submerged aquatic vegetation in general, a specific invasive species, or you're trying to avoid HABs. It's really dependent on the interaction between the HABs and the SAV. You need to know what species are involved. And in-lake conditions, such as the bathymetry of the water body, the conditions in the watershed, and prevailing weather conditions will really have a huge effect. We've been seeing over the last, let's say, 5 to 7 years, how climate change and extreme weather events have really had an impact on the ecological interactions within lakes. So both in-lake and watershed actions are required for the long-term protection of any lake ecosystem, and this includes the protection from invasive species. So thank you for your time.
Excellent. Thank you so much, Fred, for sharing your insights on those outcomes of management strategies and how those might influence the system. And to all of our presenters for really covering such a wide range of this issue. And obviously there's a lot we still have to learn, but I think this was a great overview. And unfortunately, we've run out of time for questions. I think we were able to get to all of those in the chat, and some that came in via email have been answered by Stephen and Fred in their presentations. So if you do still have any burning questions, please send an email to nisri@cornell.edu. We're happy to connect you with our experts. And I wanted to again thank everyone for being here today for New York Invasive Species Awareness Week. Keep an eye out for other events in your area on nys.info/New York ISAW. And with that, we will conclude today's State of the Science webinar. Thank you, everybody.
Thank you, everyone. Have a good weekend.